Hi, everyone. Good morning. Thank you so much for coming in today. I have a few announcements and then I'll introduce our speaker. First, if you need CEUs, make sure you sign this separate sign-in sheet before you leave. I won't be able to give you any CEUs if you don't sign it. Second, our next meeting after this that I wanted to get on your radar is a meeting about weeds in Escondido. You'll check that out. And now I'd like to thank our sponsors for making today possible. So thank you to Brokaw, Westphalia, Del Rey, McDaniel Fruit, Nutrient Ag Solution, Westpac, AgWest, Index Fresh, Mission Produce, PhosGrow, and then the Avocado Society, the Avocado Commission, and UC. Today's really, really special because we have Professor Mark Hoddle coming to speak with us. He is a faculty member at UC Riverside and the director of the Center for Invasive Species Research. He's an expert on biological control of invasive pests. And some of the pests that he's worked on include ants, glassy wing sharpshooter, ACP, and other citrus and avocado pests. He's received awards and honors from places like DPR's IPM Award for ACP control, the Avocado Society's Oliver Atkins Award for Research Excellence, and a lot more. And today he's going to tell us what we know about avocado lace bug. The goal of today is to really hear from you. So after we have our lecture from Mark, we're going to have a panel come up here. We'll have time for a Q&A. And we really want to hear from you. What should be our research goals? What should we be focusing on to enhance control of this really challenging pest? So please help us by engaging today. And with that, thank you, Mark.
All right, thank you. And I appreciate everybody taking time out to come to this meeting today. I know this is a bug that's been causing a lot of concern, and it's one we've been dealing with since about 2004 in different parts of California. And I'm going to be leading you through the initial invasion of this pest into Southern California in 2004, and then the steps that we've been, following together as it's spread throughout the state. And now it's, well established up here in the Carpinteria area as well. So on the slide, you can see some of the leaf damage. I think this has a laser pointer on it. Yes. So these are bacon leaves down in, Chula Vista. Okay, can I go backwards? You can see the egg trails here that the adults will put out on the leaves. And yes, we see a little bit of biological control through some of these generalist predators like this lacewing larva here. But I'm going to temper all of this by saying, and I think most of you have a pretty good sense of this right now, is that our control options for this bug seem to be pretty limited. And if you're relying on natural enemies to provide a lot of pest suppression, you're probably not going to be getting the levels of control you would like to see. And there's several reasons for that, and we're going to get through those in this presentation this morning. But I completely agree with Hamatal. I think what we need to get out of this meeting more than anything else is not me standing up here regurgitating all this information that we've been generating over, a long time now. But we need to get a better feel for what you guys are dealing with in the field, how you would like to see a proper, I consider myself a blue-collared entomologist. I don't do a lot of fancy stuff, but we need to come up with pragmatic, applied solutions that are going to help manage this problem. That's what we're hoping to get out of this today with your input. I can sit in the lab and in the office thinking, we need to do this and that. I'll get some phone calls. Maybe I talk to Tom and Jim and some other PCAs, Jane, and get some feedback, but it's not enough. We need to get a really detailed inside view from you guys who are dealing with us in the field on pretty much a daily basis. Okay. So what are we going to go over? We're going to go over a little bit of the history of this bug. And I think what's very interesting for me, and I'm going to explain to this as we go through the story, is that this insect was first described in 1908 from Florida infesting avocados. And since that time, this insect has always been considered native to the Florida region of the United States. And this is probably not correct. Way back in 1908, the molecular data has suggested to us that this insect was actually an invasive problem back then, but we didn't have the tools to understand that at the time, and we're going to come back and revisit that a bit later on in this presentation. Okay, so if you consider Florida to be the native region, which it's not, but if you do, just to get the story going, somehow in 2004, this insect showed up in backyard plants in Chula Vista and National City. And it stayed there for a very long time. It didn't really move out of those urban areas. And that strain that invaded in 2004 had a strong association with avocados that were related to the bacon variety. It didn't seem to infest Hass at all. So very localized, very restricted to the bacon type varieties, relatives of avocados. In 2014, it showed up in Portugal. 2017, I got calls from PCAs in Temecula, Fallbrook, Bonzel, saying, hey, avocado lace bug is here. And I said, I couldn't believe it. It'd been 13 years and it hadn't moved out of Chula Vista, and now it's shown up in Hass, commercial Hass orchards, in Bonzel, Fallbrook, parts of Temecula. So I didn't discredit these guys, but I had to go out to the field and take a look myself. And of course, they were absolutely right. Enrique Ferro was part of this group leading this charge at that time. It's like, wow, what's happened here? Something has changed. It's gone from being a bacon-liking pest confined to backyard plants in Chula Vista National City, now to expanding its way through commercial Hass avocado orchards in commercial production areas. What changed over that 13-year period? Then it's continued to move. 2019, lace bugs somehow got to Hawaii, and we'll talk about how we figured out how that probably happened. 2022, 23, more or less, it showed up in Santa Barbara, Orange, and Ventura counties. And now, amazingly, if anybody can give me any insight into this, how did it end up in Gabon, Africa in 2022, infesting avocados there? I think there's one commonality in all of this invasion history, is that people are moving infested plants. That's the only way this insect can move around. It's not a strong flyer. There's no way it could fly into the parts of Africa and infest avocados there. It's almost certainly came into San Diego through infested plants, and we've got molecular evidence to support that story as well. So this just hammers home the point. Persea mite, avocado thrips, red-banded whitefly, avocado lace bug, they are all leaf-feeding insects, and the only way they could have got here into California is through the illegal movement of infested avocado plant material that was probably smuggled into the state. That's my firm opinion on that. Okay, so adults and nymphs, they live on the undersides of leaves. You've seen the feeding damage. It causes that sort of necrotic, bronzing, almost like a burn-like appearance that starts about the middle of the leaf and then starts spreading as these colonies expand the territory that they're colonizing on the undersides of the leaves. And of course, as with Persea mite and avocado thrips, which also live on the undersides of the leaves, this makes management difficult because if you're going to be using insecticides, you've got to get it underneath the leaf, get good coverage, and that's going to be a challenge in some of these orchards where trees are tall and the canopies are interlaced. So host plants in here in California, we mainly see it restricted to avocados, but in Florida it's been recorded infesting camphor and red bay. But the commonality of all those host plants is that they're all in the same family, Lauraceae. So it likes avocado and avocado-like relatives. Okay, so the life cycle is pretty simple. Females will lay their eggs on the undersides of leaves. Those eggs, as you see those long, black, greasy, tar-like trails that they're laying on those leaves, those eggs will be laid individually in lines, sometimes singly, almost always with a pedicel, a little stalk that the egg is attached to. Then there are 4 nymphal instars. So when the eggs hatch, the nymphs will move through 4 stages. They shed their skin each time they move to a later stage. Then in the final stage there, you can see now that they're beginning to develop their wing buds. And then they'll just go through one more molt, which is kind of a dramatic change into the fully formed adult with these beautiful lacy wings. And that's how it gets its common name, lace bug. It's from those wings with a beautiful venation on it that sort of looks like a lace doily your grandmother or my grandmother would have crocheted and had scattered around the house on just about any flat surface she seemed to be able to find to put them on. Okay, so life cycle is super simple. It's on the undersides of the leaves. Males and females mate, obviously. The sex ratio is approximately 1:1. Those eggs get laid on the undersides of the leaves. The adults will live for about 23 days. In the lab, they'll lay average about 51 eggs over that life period. The eggs will hatch after about 9 to 10 days. They really like the warmer temperatures, around 90 degrees Fahrenheit, 85 to 90. Once you get above that, 90 degrees, they really don't like the heat. There seems to be an inflection point for heat tolerance at that stage, above which their populations will crash. And we've seen that in the field when we have these prolonged days of high heat, and if it's accompanied with these Santa Ana winds, the populations don't like that. We have seen it just based on the field distributions where lace bug densities tend to be lower in these internal hotter areas where avocados are growing. The populations seem to be more abundant and the pest does better in these cooler coastal climates where the marine influence can sort of like take the edge off those upper temperatures. And I think that's what we're seeing up here in the Carpinteria area, and we're seeing it down in Oceanside in San Diego County as well. They'll spend about 11 days in the nymphal stage before they complete that hemimetabolous pseudo metamorphosis that they undergo to develop their wings and they can move around. Okay, so even though this insect has wings, and it's spread, throughout California, we have very little evidence to suggest that it is a good flyer and that it will voluntarily take off and fly on its own. We've had 2.5 years of sticky card monitoring in orchards where we can have, loads of winged lace bugs on trees, sticky cards a few feet away from those heavily infested trees, and on those sticky cards we catch almost no adults. We don't understand how these things are moving around. It is probably through the movement of plants, probably hitchhikers on packing bins, maybe on your clothes, on the outsides of your vehicles that are moving between orchards as well. But we have no evidence that this insect is a strong flyer and will propel itself through the environment looking for new avocado orchards to colonize. Right, this is an unusual thing about this lace bug's biology. You will often see these egg trails. And this is very uncommon to see in these types of lace bugs. Often lace bugs will lay their eggs individually. This insect seems to engage in an egg-laying behavior that theoretical evolutionary ecologists refer to as egg dumping. And by egg dumping, what happens is if a female sees one egg laid on a leaf, she'll run over and lay some more eggs next to that original egg. And then more females, unrelated females, will come along and they'll start attaching their eggs to those lines that those females are now starting to spread across the leaf. And theory suggests that this may protect those eggs from natural enemies. It's safety in numbers, right? So if you are in a group and the larger that group is, the less likely it is your eggs are to be attacked by that natural enemy or killed by a parasitic, entomopathogenic fungus, for example. So I think this is interesting. It may also help to protect those eggs against desiccation. And the females tend to cover those eggs with that sticky tar type secretions, and we're not really sure what that material is, but it probably provides some protection to those eggs. Okay, so the nymphs hatch out of those eggs, they start feeding, they'll form these little swarms, they'll often stay fairly closely clumped together, and it's that aggregative feeding damage to the undersides of the leaves that ends up developing those necrotic spots that you can see on the top side of the leaf, even though all the feeding damage is occurring underneath the leaf. And I want to make this point too, and we're going to come back to it. This damage that lace bug is now causing to these leaves is compounding damage that is already caused by PCA mite and any saltburn damage that you may be occurring in the orchards that you're responsible for managing. So now we have 3 sources of leaf damage that are additive now, and that's increasing the damage levels to the leaves. And I don't think you need to do a lot of experimentation to probably come to the conclusion that the more damage you have on your leaf, the less photosynthetic capacity that leaf is going to have. And I guess it's not that far a stretch to assume that it's going to have a detrimental impact on fruit production, fruit set, fruit retention, and then ultimately fruit production on those trees. Now, this is interesting. At least for me, it's a very small point. But until Paloma Dadlani, a master's student in our lab, did this work, we couldn't really easily tell the difference between male and female lace bugs. But because she spent hundreds of hours at the microscope staring at adults, she determined that males tend to have these dark terminal ends to their antennal segments, but the females always have these light ends to their antennal segments. So if you're out there and you're looking and you're seeing adults and you're just curious as to what the sex is, if the tips of the antennae are dark or if the tips of the antennae are light, you can use those characteristics to distinguish between males and females. Males tend to be dark, females tend to be light. Okay, so this is some of the spectacular damage we were seeing on these bacon varieties on backyard avocados, 2004 in Chula Vista and National City in San Diego County. Complete trees were bronzed out. Looked like they'd been hit with a flamethrower or herbicide had been sprayed over those leaves. And of course, you'd end up with defoliation events. Very dramatic damage. And then if you zoom in on this, you can see these necrotic islands. And some plant pathogen specialists suspect that the damage, which I find interesting, it only seems to be associated with avocado lace bug. That leaf damage may be amplified through these generalist plant pathogenic fungi like this Colitotrichum species, which will then get into those feeding wounds and increase the amount of damage that you are seeing. Apparently, this genus is very common in avocado orchards, and under the right conditions, it can, undergo, I guess, quite an expansive manifestation when it's hooked up or in synchrony with another organisms that's also causing damage to the leaves, such as feeding nymphs and feeding adults of avocado lace bug. Okay, so we're going to come back to this temperature thing now. So it's quite hard to run temperature type experiments in the field. So what we do is that we set up these temperature cabinets in the lab. We have 3 of these. And typically what has been done, if you wanted to study the developmental biology of an insect at a temperature range that you might see in an avocado orchard or through an avocado growing range in California, you'd set the cabinet to say 25 degrees or maybe 76 degrees Fahrenheit. And you just see how long it takes the lace bug to develop at that temperature. Well, that's very unrealistic, right? Because nowhere in California where you grow avocados are you going to have a constant 76 degrees in the morning, mid-morning, afternoon, nighttime, in the very early hours of the morning, right? Temperatures fluctuate all the time. But over the course of a 24-hour period, it may average 76 degrees Fahrenheit. So what we do with our temperature cabinet studies is that we gathered massive amounts of weather data throughout the avocado growing areas of California. And if we wanted to have an average 76 degree Fahrenheit temperature, we scanned those big data sets, looked for those averages that reach 76 over a 24-hour period. We then looked at the hourly incremental changes in those temperatures, and we programmed those temperature cabinets to ramp up, then ramp back down. So over the course of a 24-hour period, you'd have a lot of temperatures that were below 76. You'd have a lot of temperatures that were above 76. But when averaged out over that 24-hour period, it would reach 76 degrees Fahrenheit. This becomes really important when you are looking at high range temperatures that might be in the 90 to 95 degree region. Because over that 24-hour period, you're going to have many hours of temperatures that are going to be in excess of 90, 95 degrees. You might be at 101. You might be hitting 102 for an hour or two in the late afternoon. This exposure to that type of temperature is what has the detrimental impacts on avocado lace bug. It's that exposure to those high temperatures over the 24-hour period, even though it might only be for a few hours. Is that clear? Because it's quite an important concept that we have applied to these temperature studies here. Temperatures fluctuate over a 24-hour period in nature. We've simulated that in the cabinets. We've put those lace bugs into what we consider somewhat realistic temperature exposure scenarios that lace bug would experience in the field. Okay, so what did we find out? If temperatures are about 48 degrees Fahrenheit or lower, lace bug doesn't seem to do anything. We call this the minimum temperature. It just sort of enters a period of stasis. It's not warm enough for it to continue developing. So if it's going to develop and populations are going to grow, temperatures need to be above about 48 degrees Fahrenheit for several hours each day. Then they're going to require these degree days. So it's going to require about almost 476 degree days to develop. This is only important if you're going to be using phenological models that you can download from the UCIPM to predict what population growth is going to be like in your field, given the prevailing temperature conditions. And that's another area that we could get into later. As you'll notice, those curves, none of them are straight lines except for the very first one, but we're going to ignore that. They're all curvilinear. You go up, you hit an optimal temperature point, above which you hit suboptimal temperatures because now it's getting too hot for you, and the populations crash. So we've determined, based on these models and what we've seen in the lab, that this lace bug does best when temperatures are averaging around 88 degrees Fahrenheit. Excuse me. Okay, so once temperatures get above 95 on average, the lace bug does not like it, and the populations will probably crash. But you're going to need those temperatures for several hours a day. So when we have hot days, strong Santa Ana winds with low humidity, that is going to be very bad for the lace bug. And we've seen these results before with avocado thrips, for example. They don't like those high temperatures, and that's why some areas tend to be more infested than others because of the average year-round climate that those orchards will be experiencing. Are there any questions on that before I move on? Yes, down the back.
Question is, based on what you're learning from this, down south, we're seeing a lot of adults overwinter.
Yeah.
Do you think this slowdown model is contributing to them being around in the environment longer?
Yeah, right. So I think temperature, like you said, when they drop down, they'll live longer because insects, their longevity is going to be affected by temperature. On average, the warmer it is, the more, I guess if you want to look at them having a finite time span, it's temperature driven. So the warmer it gets, they're going to be sort of like more active and they're going to burn up their longevity credits or their lifetime credits if you want to look at it like a video game. So yes, if it cools down, they're going to live longer. And the other thing with what you're observing down there, and we're going to get this a bit later, is also related to leaf drop on the trees. Because if you have trees that are retaining leaves for a long time and those leaves are supporting lace bugs, you're going to have more abundant populations on those trees. When we go through these defoliation periods, I sort of think of avocados as being semi-deciduous. You have defoliation periods. A lot of those lace bugs are going to drop to the ground on those older leaves. Then there'll be a period where the temperatures may be cooler. The new leaves are flushing out. You think, oh, I don't have much of a problem right now because the percentage infestation of those new leaves that are coming onto the trees don't tend to have a lot of lace bugs on them. But the leaves that do have lace bugs tend to be more highly infested on average than what you would see otherwise because there's a less of a resource for them and they're concentrating on those few leaves that they can get themselves onto. All right, so let's just summarize this, and this is a bit of a complicated graph. So when temperatures get back to this point, when temperatures are on the cool side, lace bugs are going to live longer. When the temperatures are getting hot, 95 degrees Fahrenheit and higher, they're not going to live for so long. And you have multiple days of prolonged high temperatures and low humidity, populations are going to probably, I wouldn't say collapse, but decrease. Temperature also affects the amount of egg laying that we see. So this is interesting because this becomes a double whammy for the lace bug. There's going to be an optimal temperature range over which it can live and lay eggs. And what we have seen is that when temperatures are on the high side, they'll lay a lot more eggs, but they don't live as long. And then the flip side of that is when it's cooler, they will live longer, but they won't lay as many eggs. So there's a balancing act that this lace bug is facing every day that it's in the environment between temperature and how well it's going to reproduce. And again, this also affects the fertility or the percentage egg hatch. It's going to be a balancing act between optimal temperature, which is going to affect the amount of eggs that it lays, how long it's going to live for, and then ultimately how many of those eggs are going to hatch out. So when you get to these high temperatures, like at 35 degrees Fahrenheit, for example, 95 degrees Fahrenheit, the eggs just don't hatch. They're getting cooked on the leaves. The adults aren't living as long either. So all this evidence is supporting this idea that as temperatures go up and it gets hotter and drier, lace bug doesn't like it. which it probably also suggests that it's come from parts of Mexico where it's native that tend to have cooler year-round temperatures. It's not a hot, adapted species. Okay, so we've tried to capture this in this somewhat complicated graph, but I think what we have generated here, the take-home message is, when we were doing these surveys down in San Diego County, we had 2 orchards in the oceanside area where it's cooler and the marine influence is affecting lacebug temperature exposures. And then we had hotter interior orchards in the Bonsall zone, which tend to escape the marine influence somewhat. So what we have noticed with that is that during the fall and the winter, the percentage of leaves that will have lacebugs on it tend to be higher. But the densities tend to be greatest through June through October. And this is a resource spreading outcome that we see in the orchards. When you have lots of leaves or few leaves, the numbers of lacebugs that you're going to see on those leaves is going to be proportional to the amount of foliage that is available to be infested. So when these trees start going through a defoliation period, you're going to have fewer leaves, but you're going to see higher numbers of lacebugs on them. As the tree canopy fills out, you're going to have more leaves. The lacebugs are going to be spread out over more of those leaves, but the densities of those lacebugs on those leaves is going to be greater. So this is something you might want to keep in mind with your sampling, is the time of year, what the phenological state of the tree itself is, and the temperature exposure that those populations are going to be experiencing. But the take-home message overall is that if you're in an interior area where it's hot and dry, lacebugs tend to be lower. If you're in a cooler area closer to the coast, lacebug pressure is going to be greater for you. Okay, so there appears to be some cultivar differences in susceptibility to avocado lacebug too, at least this new one that we're dealing with. The very first one that came into California in 2004, it really liked bacon and bacon-type relatives. Now we're seeing, since 2017, the new lacebug really likes CAS. But the orchards that we were monitoring, we could also look at reed, zutano, gem, and fuerte. So reed also had quite high lacebug levels on it. Zutano was similar to reed as well. Gem, which is becoming a more popular commercial variety, lacebug didn't seem too interested in infesting those gem trees. But fuerte, which was really quite interesting, as its name would suggest, was really quite resistant to lacebug. Over the course of 2.5 years, the fuerte trees we were monitoring, even though they were very close proximity to the house, they were pollinators they had out there, we didn't see any lacebugs on fuerte. So, if there's going to be any plant breeding efforts, there's some clues there as to which parents might be good for generating new varieties that could be potentially resistant or tolerant to avocado lacebug. Okay, natural enemies. We have a lot of generalists that live in our avocado orchards. A pretty important one for us are these predatory thrips, which are in the family Aeolothrippidae. We have 2 genera which you'll find. The one that you see up there right now is Franklinothrip. The other genus you'll see is Aeolothrips. They're both referred to as banded-winged thrips, as you can see, for various reasons, that they have those nice striking bands on their wings. You'll see green lacewing larvae out there as well. We saw a lot of whirligig mites eating things on the leaves. Jumping spiders were quite common too, and we saw a lot of ladybugs. Okay, so given all the generalist predator activity that you see in orchards, is there any evidence at all that any of these things are providing any sort of pest suppression, population level control? 2 guys down there, Jim and his neighbor, shaking. No, there's nothing. And this is what we saw in 2004 too. And when Phil and I did all the foreign exploration through the Caribbean and other areas where lacebug numbers were really high, we found no evidence of any of those populations that we were surveying being regulated in any way at all by any type of natural enemy that we could bring back here to California for use in a biological control program. There's possibly one exception, and that's this guy, Franklinothrip rhizobensis. You may be familiar with this because this predatory thrips is also quite important when we look at avocado thrips populations. That seem to be this thrips, and it's not congener, but the other one in the family, Aeolothrippidae, Aeolothrip kawani, These 2 species of banded wing thrips that you have in your avocado orchards respond quite strongly to avocado thrips outbreaks. This ant-mimicking thrips that you see up here, Franklino thrips, may have a somewhat weak response to increasing avocado lace bug populations, but it's not sufficient enough to provide any reliable or give you confidence if you see it out there that, yeah, this thing's going to drive these numbers down to low levels and I don't need to worry about it. We don't have that luxury with any of the natural enemies that we have surveyed for in these orchards. Okay, so insecticides right now tend to be perhaps our most powerful tool that we have for managing avocado lace bug. And Tim Spann prepared these nice insecticide tables, which he published in an article that's, you may have, how many of you read the From the Grove that the Avocado Commission put out? So great. So you may have seen this because I basically ripped Tim's tables out of his article and I put it up here so we could go through them. There's the web address, but just type in avocado lace bug from the Grove and it'll bring it up in Google for you straight away. Okay, so there are a bunch of insecticides that are currently registered for use on avocados, and they are registered for use against avocado lace bug. And I'm not going to go through all of these. You take a photo of that, check with your PCA as to what's available, and then make a decision on what may or may not work for you. But this is what I want to point out on this slide. So there is a scientific group that's very interested in maintaining the efficacy of insecticides. And it's called the Insecticide Resistance Action Committee. And they look at all the insecticides that are commercially available, what their mode of action is. For example, is it a gut poison? Does it affect the GABA channels in the nervous system? Is it an acetylcholinesterase inhibitor? And based on the actions of those insecticides, they put them into these different classes. And you can see here, we've got classes 3, 6, and 4. But what I want to highlight here is that a lot of the insecticides that are registered for use against avocado lace bug fall into class 3. And the IRAC committee specialists say this is a bad thing. If all of your insecticides are in the same class, even though they're different product names, there's basically the similar active ingredients acting on the same parts of the insect that are killing it, it is probably going to develop resistance. And then you'll end up with resistance to multiple products that have the same classification. So we have a problem right now. We have a bunch of things that are registered, but they're basically all the same, if you want to look at it from a mode of action point of view. So overuse of those insecticides will probably lead to resistance development, and then you will have nothing available to control avocado lace bug. So this is where you need to think about judicious applications, insecticide rotation schedules where you rotate insecticides with different modes of action so you're not exposing the insect to the same chemical class over and over and over again. So I want to give you an example of this where it was really highlighted to me. I was working in Saudi Arabia for several years on red palm weevil. And dealing with the date growers over there, they're saying, oh, and none of our insecticides are working anymore against this red palm weevil. I said, that's interesting. I'd like to know more about this. Provide me a list of all these insecticides that you're using. They had a table about, I think it was 15 insecticides. They go, look at all the insecticides we're using. How could it possibly have developed resistance to these things? So when you looked at the IRAC classes, they're basically all the same. What they had thought they were doing for an insecticide resistance management program wasn't based on IRAC class. It was based on the name of the insecticide. They thought rotating through a schedule of insecticides with different names was a good resistance management program. But the reality was they were rotating through insecticides with different names, but they all basically had the same mode of action. That's why they ended up very quickly getting resistance to those insecticides, and red palm weevil flourished. Okay, so realizing this, the Avocado Commission has been working with the feds. We're looking at this IR4 to get additional insecticides registered for use against avocado lace bug infesting avocados in California. Okay, so right now, of the 8 insecticides that have been looked at, 5 are registered for use on avocados, but the label doesn't cover avocado lace bug. An additional 3 products are not registered for use in avocados, but they are being looked at. There is one group there that really worries me, and it's this one, isocycloserum. How many of you have heard of these forever chemicals, these PFAS chemicals, right? Yeah, they're in these fire retardants. For example, they get sprayed on kids' clothes so they don't burst into flame should they run into a flame, or they cover up your couches and all that sort of thing. So if you fall asleep smoking a cigarette, you won't incinerate yourself on your couch. These chemicals are so long lived in the environment because they're stabilized with these halogens like chlorines and bromine molecules that they basically never degrade in the environment. And we all have them in our bodies right now. We're all loaded with these forever chemicals. So I think it would be a very bad idea to register a forever chemical that's an insecticide that's going to be deliberately sprayed on our food. And I hope this one doesn't get considered for use in California avocados. Okay. So the other thing to consider here is if you're going to be applying insecticides, probably high volume sprays are going to be required because you're going to have to get underneath the leaves and you want very good coverage on the undersides of those leaves. So aerial applications may be difficult if your trees are big and the canopies are interlaced. However, when we were doing some of this work for avocado thrips with Phil, Ben, and Wee Lee, was it Wee Lee? Yeah, way back in the day. Phil was kind of high on the helicopter thing because if the helicopter could come in low enough, and he called it prop wash, where the canopy would be highly agitated and you could see all the spray going up underneath that canopy as the prop wash from those rotating blades was really agitating the canopy. That could help increase coverage. However, the recommendation, I think, is to make sure that your trees are well pruned, the canopies aren't interlaced, and the tree height is somewhat low. So it's also been pointed out that if you have these high-density plantings or you're growing dense hedgerows and they're infested with avocado lace bug, getting sprays into those types of canopy situations may be quite difficult. Okay, so let's get back to leaf damage. Right now we have 3 types of significant leaf damage in avocado orchards. Tip burn from the accumulation of chloride salts in the terminal ends of the leaves, which occurs through salty water applications. Been dealing with PCA mite since about 1990, and you can see all the little necrotic spots that form along the midrib and the lateral veins. And now we have avocado lace bug feeding damage adding to that. Okay, so one of the questions we were interested in answering is, how much of your leaf is being destroyed now through salt burn, PCA mite feeding, and avocado lace bug damage? So it can be quite hard to measure these leaves, but now we've got some very cool tech that's freely available on the web, and we use this software called ImageJ, where you can basically take a digital image of the leaf, program the software to look for PCA mite damage, tell it what salt burn damage looks like, and then tell it what avocado lace bug damage looks like. And it can rapidly scan through hundreds and hundreds of these digital images. So we pick these leaves from the field, bring them back, put them on a flatbed scanner, get the digital images, ask ImageJ to look at it, classify the amount of damage based on PCA mite feeding, avocado lace bug feeding damage, and leaf burn. Okay, so what have we found out from that? Well, between September and November, at least in the Bonsall, Oceanside area down in San Diego County, leaf damage is going to be ranging anywhere from around 1 to 16%. Fall and winter, about 46% of your leaves are now going to have lace bug damage and PCA mite feeding damage on it. But in spring, as the new leaves are coming out and the old leaves have been dropped, those new leaves are obviously going to be clean and they'll have very little damage levels on them. Okay, so what we have found is that in most months, now the cumulative damage load on those leaves is going to be 10% or more, and occasionally it's going to reach at least 20%. I think when you're getting up to that upper level damage, I'm unaware of any scientific data to support this statement. I just don't think it's going to be good for the tree. If 20% of your leaf is now being damaged through a combination of these 3 events, PCA mite, salt tip burn, and avocado lace bug feeding damage. So the other thing is lace bug, as you've seen from those necrotic spots, on average will be doing a lot more damage to a leaf than all of the cumulative damage from all those little nesting spots that PCA mite puts on the undersides of the leaves. So this may be another reason why lace bug control should be considered to minimize leaf damage and possible photosynthetic reductions, which may lead to reduced yields. Okay, right. Any questions on this before I go into the invasion history of this bug? All right, still awake. Guess we've got one question. At least one person's paying attention. I'm getting hot up here too.
In my experience, it seems that the ALB leaf damage causes defoliation, whereas PCA mite damage can cause defoliation, but not as significant. And then my question about that is, what happens to the ALB when it hits the ground?
Yeah, so that's a good question. Okay, so yes, PCA mite can cause defoliation. And now I'm going to show how old I am, because when I got here to UCR back in the day, it was when PCA mite was raging full on. And defoliation, Jim probably remembers, and Tom probably remembers. You don't remember too long ago? It was bad. Trees defoliating. Lace bug, yes, can certainly do this too. I think whatever's happened now, PCA mite doesn't seem to induce those defoliation events as frequently now. Is that, or maybe even at all? Is that a fair comment?
Not high enough, but we basically don't let them get that high.
Right, okay. So management is keeping it below a defoliating density. Yep. So yes, so possibly the same with avocado lace bug. Another incentive for considering control will be to minimize defoliation events and to minimize damage to those leaves, which will drive down photosynthetic activity too. So what happens to lace bugs on the leaves when they drop to the ground? We don't know. Same thing with PCA mite when they drop to the ground. We don't know. We did see way back when we looked at whether or not they would leave the leaves and migrate and go back up the trunks. We saw no evidence for that with PCA mite. But whether or not lace bug, which is a bigger insect, can walk far faster, it's got longer legs, maybe it does that type of thing. Maybe that's when it flies. It's just vertical flight rather than horizontal dispersal flight. So that basic question we got no answers to right now. Yeah. Yes.
Can you share a little bit more about the IR4 process and where it's at? Is your lab currently evaluating those products? Are those field trials, lab trials?
Oh, yeah. So all that IR4 stuff.
Yeah, the 8 products.
Yeah. So my understanding, based on a very brief communication with Tim Spann, is that the Avocado Commission has been working to get this done. And I don't know, Bo or you or me, we haven't been asked to help with any of that. So basically I just took that from the Grove, and Tim said the commission was working with the feds to get this done. And I guess the nice thing about this is that if your products get selected for evaluation, the federal government will pay for those evaluations, so it doesn't cost the industry anything. So that's probably why they've gone that route. If that doesn't pan out, then they might be asking us to look at it for them. Yeah. Yes.
We got hit really hard, a lot of defoliation. It seems like on a lot of them, there'll be just a spot like as big as a dime and the leaf will fall off. Doesn't seem like it was decreasing the photosynthesis that much. So
Right.
you spoke to that before about a fungus or something that might
So there's
affecting that.
Right. So there's this genus Paleotrichum, which seems to be quite common in these avocado orchards, which, from my understanding, talking to some of the plant pathologists at UCR, will take advantage of damage to leaves, for example, and maybe fruit too. I'm not so sure on that. So if you have a lot of lace bug feeding damage on the undersides of the leaves, it creates these entrance wounds for the fungal spores to get into. So what you're seeing on your leaves is probably a mixture of lace bug feeding damage, which probably is getting amplified by a secondary pathogenic infection from these Paleotrichum fungi, which obviously, I guess, will probably increase the amount of damage that leaf suffers. So when you have small spots on your leaves, it's probably not so much of a concern. But when you have lots of small spots over many, many leaves, that's probably when you start to see the damage levels starting to increase in a cumulative way across multiple leaves rather than a few leaves having a lot of damage to them.
A leaf drop on leaves that only had a spot
Right.
the size of a dime.
Okay.
So it wasn't like, I mean, some places it spread, but because we're
Yeah.
looking at it like, that's not too bad. It can't be affecting the leaf that much. And then the leaves fell off.
Right. Okay. So that's very interesting. And maybe some other people have comments on this. You know, it's possible that the tree is reacting to an infection that it's really quite sensitive to. And there's been a lot of speculation in the industry that Persea mite damage and I think avocado thrips damage to leaves when it was initially a new wave of infestations coming through. Plants seem to be very sensitive to that damage and would drop the leaves. Would that be a fair summary? And then through time, anthropomorphizing here, the trees sort of like got used to it and could hang on to their leaves. So maybe with lace bug, what you're seeing is similar to what we saw with Persea mite and avocado thrips, is like an alarm response by the tree to damage that it hasn't been subjected to before, And so what causes that? Well, it may be salivary type enzymes or toxins in the lace bugs saliva as it's starting to feed, which could be creating, if you want to think of like an immune response, causes the tree to drop its leaves prematurely, even though the amount of damage to that leaf probably seems small and an unjustifiable response by the tree. I don't really know, actually. Yeah.
Yeah. Oh, thank you. Yeah, I'm from Carpinteria.
Okay.
And of course, we've had the warmest winter in over 130
sure.
years up there this last winter. So, I mean, I'm sure some have overwintered, but I don't know how they're overwintering. I think once they hit the ground, I think that's it. But we did pull the trigger in November, and we did spray Mustang and Meyer and some oil on them.
Right.
And that seemed to stop them, but it seemed like I was noticing most of the populations about a little bit head level down to waist level, and in the necrotic areas were difference in size. So at that time, it seemed like it was spreading, and it was starting to spread out in my PCA said it was spreading out in Gubernador Canyon. So he started in Gubernador Canyon, and where they had a lot of defoliation out there. When you get the defoliation, you got the exposed fruit. Of course, it doesn't go to the fruit. Right. And then all of a sudden we get hot days like we've had here in the last few weeks. Now we're seeing the sunburn on the fruit, and which is not going to be harvestable. That's going to be nothing. So what we did, we kind of stopped it at that point with the combination of the spray. And so it did stop it, but we still had the defoliation was going on. I noticed that in areas where I had stronger trees, I didn't have that much defoliation. Where I had weaker trees, I have a 10-acre block, which we were waiting to harvest. We was hoping the price would go up, and of course the price went down, and we got hammered on that. And so it's economically, it's been a hardship for us because of this thing coming in. And we've had to go with an extra spray. Usually we do one spray a year for the skirtle thrip, and that seems to do it with the Agrimec. And so we do that and get it done, and then we kind of coast through the rest of the year. We do have some nesting whitefly going on, but other than that, the colder winters will kind of stop it. And now we're coming into spring. I did do an inspection out there. I was looking for some more lace wings, and of course they were. You look at where the necrotic areas on the leaf, and you pull them over, and you see that they look kind of like a black varnish area in there, and it's kind of sticky. And I was wondering too on if you had some sort of biological activity or predaceous insects, would they kind of not go to those sticky areas?
Right.
And
probably not.
And that way it would kind of repel them, and then we wouldn't have good control as far as biological control.
Right.
So that would, so we got to find something that's going to help or bring something in that would help. Also, I noticed after the spray, and I used Mustang, was a pyrethroid. My brown mite is starting to flare.
Yeah.
So I've gotten a flare now on that. So I'm not happy with that. So I've got to deal with that.
Yep.
Then I was going around and looking at all my new flush. So everything's flushing right now pretty good. But then I got the omnibus relief roller in there happening. So I mean, we're getting hammered from different things.
Right.
So I kind of see that's what's going on.
Yeah. Yeah. So some of that might be insecticide driven, but I think you made a couple of interesting points here. So it seems based on spraying, say using a product like Mustang, even though the leaves were sprayed when there was a small amount of damage, it was still the spray. I guess you consider the spray was still applied too late to stop the defoliation, even though damage levels were small. And then what's a weak tree? Is that a Phytophthora stress tree? What's weak? When you say you got weak trees in your orchard and you see intense lace bug activity on those weak trees,
we do treat for Phytophthora using Ardonis. We use that. We put the Ardonis in twice a year.
Okay.
So we do that. The soil treatment, we put that in. Like I said, I was holding fruit on that. That's where my biggest damage was, and so we completely sprayed that 10 acres. On the other 30 acres, I did a perimeter spray because I was going inside. I didn't see any damage on the interior. So I think they were moving from the edges in.
Possibly.
Now, I was out with my PCA. We were looking at it, and before we sprayed, we're doing an inspection. I had a powder blue sweatshirt on, and they were all over my sweatshirt. They were traveling with me, and they were on him, and so we're kind of brushing them off. So they were hitchhiking. Those things really are strong flyers.
I don't know if there's so much strong, but we just don't have evidence for that based on our, walking through a canopy and having things drop on you, brushing, maybe short distance movement from a tree onto your sweatshirt, whether it's blue or yellow. That's not really the same as high, I'm going to fly 3 miles to get myself to a new orchard. See, so with Persea mite, for example, they have no wings and they can't fly. But they are more than capable of spreading many, many hundreds of yards. When you have a very high infestation of Persea mite on a leaf, when the temperature and the wind conditions are right, you will see those mites go to the margin of that leaf. They will then put out, I've got photos of all these silk strands. And then once the wind catches that silk strand, the mite just releases itself and it'll blow downwind a really long time. So even with prevailing winds, we still see no evidence for avocado lace bug dispersing like this.
My the Hass on Torals were mostly affected.
Yep.
I've got gyms up on a hill.
Yep.
Now the gyms, there was nothing on my gyms.
Yeah, that's good. That supports what we've
Yeah, and I saw your chart. So really, the gyms are actually pretty good. And so we didn't see that. I have other things happening on the gyms. And when I go to my trees, they're healthier trees. I didn't see very, very little damage at all. So the stress trees seem to have the most damage. The stress trees.
So I think we need to make a note of this because this is one of the things we come back to as, research priorities. But we probably should start pushing on. Well, this whole questions will come back and put this into the discussion. Okay, so thank you. Interesting points. Good foundation. I see you guys taking notes.
Mark, we've got several Zoom questions
Oh,
here.
we do? All right.
I'm going to bring them all together.
Yep.
Do you see any damage, branches?
No branch damage.
Are you seeing as much damage to young leaves as old leaves?
They prefer older leaves.
And with the secondary fungal infection, should people be applying copper as well as an insecticide?
Okay, if you're going to try to manage lace bug, I would go after the lace bug and not the fungus. So I would prioritize an insecticide application over a fungicide. But I'll combine both of them. I don't know. Any thoughts on that? Jane Delahoyt is saying no. All right, that's it. Case closed. Move on. Jane says no. Okay, so 2004, as I mentioned, we first found avocado lace bug in people's backyards. Did really well on bacon variety, relatives, didn't spread. The DNA told us that population had originated from Mexico. Okay. 2017, we start getting reports of lace bug now in commercial Hass avocados orchards. So what happened in that intervening 13-year period? Why did it go from being a bacon specialist in people's backyards to now a Hass specialist in commercial orchards? So the DNA told us something had happened. There had been a second invasion of a different type of avocado lace bug into California. First one in 2004 came from Mexico. The 2017 population, when we finally got around to looking at it, had come from somewhere else. The DNA suggested it may have come from Florida, which if you cast your mind back, that's where lace bug was first described in 1908. And I think we have, and it is always assumed to be native to Florida because of that very early description of that species. But the DNA analyses that we have done strongly suggest that Florida was invaded a long time ago by avocado lace bug, probably from populations that originated on the west coast of Mexico. And we're going to get back to that in a minute. Okay. 2019, LA County now has reports of avocado lace bug. Interestingly, on bacon-type trees growing in people's backyards, which strongly suggests that the original San Diego population somehow got moved into LA County. How that happened, I don't know. And then 2019, Hawaii, reporting massive outbreaks across 5 islands. The DNA analysis suggests that the avocado lace bug that invaded Hawaii had the same genetic fingerprint as California, which is the Florida/Caribbean variety of avocado lace bug. So how did Hawaii get avocado lace bug? Did it go directly from Florida? Or did we, California, send it to Hawaii? I'm going with California sending it to Hawaii because we're much closer than Florida. 2022, Orange County invaded. 2023, Santa Barbara, possibly Ventura counties invaded. 2000, who knows when, will San Luis Obispo end up with this problem? Any reports from SLO? No, not yet. Probably just a matter of time, I would think. There weren't, but I just assumed that to be the case. And is that incorrect? It's not here. That's probably why I couldn't find anything on the web. Okay, I need to change that. I should have called Tom. So it's only in Santa Barbara. It's not in Ventura. Okay, so yes, but not really. Okay, all right. Okay. This is where we thought avocado lace bug was native to, based on that original 1908 description and the fact that lace bugs have been collected all through this, Pacific, general Caribbean type area. So Phil Phillips and I, with funding from the Avocado Commission, to deal with this 2004 invasion, there was a lot of interest in biological control. Could we find a parasitoid, especially one that would go after the eggs of avocado lace bug, that we could bring back here to California to use in a biological control program targeting this pest? So since it was assumed that lace bug was native to Florida and the Caribbean, we spent a lot of time going through these areas collecting thousands and thousands of avocado lace bug eggs. And the assumption was, if there's a good natural enemy there, I should be able to rear it out of those eggs that we've collected all through the presumed native range of avocado lace bug. Here's Phil, St. Thomas, doing the same work that I'm doing. Okay, so now we had all these samples and all these eggs that we'd collected from the Caribbean. We also did a lot of collections, as you can see, down the Pacific side of Mexico. That had never been done before. Until we did this work, avocado lace bug wasn't known from the Pacific side of Mexico, which was really interesting to me. And I wasn't expecting to find it, but just to do our job properly, we went down into those areas to look anyway. So we got samples through Guatemala, even down into South America. So what we were interested in was, could we get a DNA fingerprint for California's population and match it to some of these populations that we had sampled throughout all these different countries? And the other question we wanted to answer was, Is avocado lace bug one species, or is it a bunch of species that we can't tell apart morphologically? We refer to these as cryptic species, and we deal with this a lot in entomology, where we have one type of organism that is, we assume to be one, but really it's made up of a lot of different species that we just can't tell apart unless we look at their DNA. Okay, so what did the molecular work reveal? To me, when these data came back from Paul Rugman-Jones, it was really quite amazing. California's population in San Diego in 2004 came from the state of Nayarit. That's a big area. But amazingly, quite by chance, I sampled avocados in a small place called Las Vivosas. And the only reason I sampled avocados in that particular area, because as Phil and I were driving back up the coast, we had to stop at a toll road. And we used the bathroom there, paid the tolls. And people had all these little tables set up along the side of the road. And one lady had small potted avocado plants in black polythene bags that she was selling. And the undersides of those leaves had avocado lace bugs on them. So I sampled those, put them in my vials and took them back. That roadside stand had pretty much an exact DNA match to the populations that we had collected from Chula Vista and National City in San Diego. So we're about, I think it was like 2,000 kilometers or something away from California. It didn't fly that distance. So what I think happened was somebody parked their RV in the same parking spot Phil and I did, used the bathroom, wandered around, saw the little avocado plants, said, best guacamole I've ever had's come from Mexico. Let's take these little plants back and we'll put them in our backyard in San Diego County. Put them in the RV, drive all the way up, go through the border. Got any plants, fruit? No, nothing. Okay, through you go. That's how easy it is. How many of you come into LAX recently? I was in there about 2 weeks ago. You know what? You don't get asked anything anymore about fruit, vegetables, carrying wood, nothing. All you do now is you walk up, look at an iPad, it gives you a green check and you move through. That's it. So, you know, with Bo's work, it's no surprise that we're being overrun with fruit fly infestation from all these different species from these many different parts of the world. There's just no border security, at least at LAX, that's of any consequence anymore. So anyway, that's a whole different story. Anyway, so lace bug, all that DNA work, it's just one species. It's not a blend of different things that we can't tell apart morphologically. There's the little roadside stand. See those little potted avocados? That's where I got my lace bugs from. Okay, so yeah, 2,000 kilometers between the sample spot and where it was first found in southern San Diego County. Somebody must have moved that plant, probably in an RV, I think, a tourist, moved it back to San Diego, planted it in their garden in a way that population went. Now, the western side of Mexico, along that Pacific coast, extremely high levels of genetic diversity in lace bug populations in that particular part of Mexico. Overall, the population's densities are low, hard to find in comparison to the Caribbean side in Florida. Evolutionary theory tells us that if you have found the true area of origin for a species, genetic diversity is high. It's been there for a long time. It's done all sorts of different things over time, you know, geographic separation, gene flow, all this sort of stuff leads to high genetic diversity. When an invasive species invades a new area, a small subsample of all that genetic diversity gets transplanted to a new area, and those invasive populations tend to have very low levels of genetic diversity. They tend to be genetically homogeneous. So when we move across those mountains into the other side of Mexico and then into the Caribbean, there are very low levels of genetic diversity in those Florida Caribbean populations of lace bug. They're pretty much all the same. So if theory is correct, those populations on the east side of Mexico into the Caribbean and up into Florida were probably invasive and had been introduced into that part of the world a long time ago from populations that are native to the western side of Mexico. We have spent no time looking for natural enemies in that western part of Mexico. And possibly one reason why those densities tend to be so much lower than what we observed on the other side of the country and into the Caribbean is because they may have good natural enemies there that could be controlling them. We don't know that. Nobody has looked, but it's a possibility. Okay, so we've got these two populations in California now. Orange one from Mexico, that blue-green one from Florida. They're both either in our backyards or they've invaded the Hass avocado orchards. 2004, the Mexican variety from Nayarit, Las Vivosas, arrived in San Diego County. It just didn't move. 2017, it looked like we got something from Florida, probably not the Caribbean. It's much easier to move a plant from Florida into California than it would be to move plants from the Caribbean into Florida. 2019, avocado lace bug found infesting avocados in Hawaii. So the question was, that Florida-type population in Hawaii, did it come from California or did it come directly from Florida? Before we did the analyses, you know, the other question was, did they actually get the Mexican variety? Or maybe they got something from somewhere else that we didn't even know about. But the DNA has told us that what's in Florida, what's in Hawaii now is pretty much identical to what we have in California, which is pretty much identical to what we have in Florida, which suggests to me the triangle probably went Florida, California, California to Hawaii. Okay, so we don't know which populations have invaded Portugal or Africa. We did look at crossing studies between the Mexican variety of lacebug and the Florida variety of lacebug. We did those crossing studies between males and females of both genotypes in the lab. And unfortunately, there is some evidence to suggest that when the Nayarit variety of lacebug and the Florida variety of lacebug mate and produce offspring, they may have something that we refer to as hybrid vigor. They do better than their parents do. So we probably don't want to be mixing these different lacebugs up if we can help it, but that may be well beyond our control right now. The other thing that was interesting about this is that we went back into those original areas of Chula Vista and National City. It's still all Mexican avocado lacebug. We saw no evidence for the Florida lacebug having pushed into those areas. Whether or not that changes over time, we don't know. Okay, and the other thing is that, you know, the Mexican variety from Nayarit seems to like those bacon type avocados, whereas the Florida variety really likes Hass. And that's without doubt what we've seen through the Caribbean, especially in the Dominican Republic, where they have a decent sized avocado industry there. It's pretty much all Hass. Get absolutely hammered by avocado lacebug in the Caribbean, in the Dominican Republic. Okay, take home messages, right. 2 lacebug invasions into California. 2004, we got that strain from Las Vivosas and Nayarit, restricted to bacon varieties in people's backyards in Chula Vista, National City. 2017, we got a Florida haplotype or a Caribbean haplotype, which has done well in California. It's spread into Hass. It's up here now in Carpinteria. It's also made it to Hawaii as well, causing a lot of damage to non-Hass varieties in Hawaii too. Okay, so temperature and development. Temperatures need to be above 48 degrees Fahrenheit for development. Optimal temperature is around 88 degrees Fahrenheit for development. Remember, that's the average over a 24-hour period. But once temperatures start getting up to about 98 to 102 degrees Fahrenheit, that's what we have determined as the upper lethal or maximum temperature for lacebug. It doesn't like those temperatures and it's going to die, or its longevity, fertility, and egg hatch rates are going to drop a lot. And that may explain why it tends to be more problematic in coastal areas where the marine influence moderates temperature as opposed to the warmer, more interior areas which are going to have higher temperature extremes. Phenology, percentage leaf infestation with lacebugs tends to be highest in the fall and winter, but population densities on infested leaves tend to be greater June through October. At least that's what we see in the Bonsall Oceanside area in San Diego County. I'm unaware of any phenological studies being done up here. Are you guys looking at it at all? No, okay. Natural enemies, I think we can write this group off. We see no good biological control in our orchards. Predatory thrips, especially frank linothrips or rhizobensis, may be doing something, but not enough to be relied upon. Leaf damage, we now have 3 significant sources of leaf damage in our orchards: avocado lace bug feeding damage, PCA mite feeding damage, and salt burn to those leaves, which together you can end up with anywhere from 10 to 20% leaf damage, depending on the severity of those 3 factors impacting your orchards. Cultivar preferences. The Mexican haplotype or variety of avocado lace bug really likes bacon and bacon relatives. The Florida variety really likes Hass, Reed, and Zutano. Gem tends to be a poor host for the avocado lace bug from Florida, and Fuerte, we saw no attacks at all on the leaves of that cultivar. All right, that's it. So I think now I'll take some questions, and then I think, do you guys
hold on, please, before you ask your questions, make sure you have, there's mics on your table, and put them on and speak to the mic, please.
Let's do questions just for like, max 10 minutes. Then we'll do the industry panel, and then we'll have more time for questions after that.
All right.
Let me go start in the back.
I'll start. I appreciate you mentioning the compounding effect of salt damage, PCA mite. We have an orchard where we have, or our orchard with the saltiest well water seems to have the lowest incidence of lace bug. Is there a correlation documented that they don't like hosting those salty leaves?
Wow, that's a great question. I don't know. I guess, yeah, I don't know. It's possible. Yeah, interesting observation, something we could look at for sure. Not that you want to be dosing your trees with salty water to control lace bugs, so I'm not sure what the trade-off there would be. Yep.
Is there any information coming down the pipeline about the use of beneficials? Like, is utilizing Rufilabris worthwhile? Because I know it's not a native species, and the lacewing that might be seen in the orchard that are feeding already could be a native varietal.
Okay. How many insectary managers do we have here? Muscle Manos. Okay. Right. I'm going to give you my very frank and honest opinion on this, and I'm going to preface it by saying my job at the University of California, Riverside, in the Department of Entomology, is a specialist in biological control. I do not recommend any grower buying natural enemies and releasing them in their orchards for the control of avocado lace bug or PCA mite or avocado thrips. And the reason I'm stating that quite emphatically is that we have done a lot of work with industry money, not on lace bug, but on PCA mite and avocado thrips, either on our own as a lab or evaluating releases made by the industry. We have zero evidence to suggest that any of those natural enemy releases has an impact. Now, I just want to dial that back a little bit for PCA mite. You can buy species of predatory mites, and we have done this, and we have put them on avocado trees for the control of PCA mite, and they do provide control, undoubtedly, equivalent to what we were getting with oil at the time, because we didn't have other insecticides or to compare against. The cost was prohibitive. We could get excellent levels of control, similar to what we could get with insecticidal oil. The cost was like, I think, 10, 15 times greater, and it required us to go out and hand place these natural enemies into the tree canopy. Tree, yeah. And then the thing with that was the timing was absolutely critical. So we determined that not based on the number of mites per leaf, but it was infestation levels of leaves. So if you were below, I think it was 33 to 50% of your leaves didn't have at least one Persea mite on them, the predatory mites wouldn't establish. If you got to 75% of your leaves or higher having Persea mite on them, there were just way too many mites for the predators to be able to bring those populations back under control. So the sweet spot was somewhere between 33 and 50% of your leaves had to have at least one Persea mite for those released predators to establish populations that would give you season-long control. The issue then became, and I still don't know why this is, those predator populations never persisted year after year in those orchards. They would disappear. So now you're basically buying a lot of predatory mites and having to put them out into your orchards again each year. We've done this with lacewings, either larvae or lacewing eggs that have been released, and we see no levels of appreciable control. So I would, when I get emailed about this, I just say, I don't think it's worth it. Yeah, and it's disappointing for me to say that, but I don't think it works.
All right, let's hear from Michelle.
Oh,
So
here comes the counter, and we're going to get sucker punched in.
here it comes.
Okay.
In the past, we've used imidacloprid in citrus, lemons in particular, for glassy wing sharpshooter and ACP. And we know now that it only translocates a certain height on the tree, maybe 4 or 5 feet possibly. And we're told that it's recommended that we don't use it for trees older than 3 or 4 years in citrus. Is there any work that's been done on avocados, because they're substantially larger, to see how far the material gets up into the tree?
Right. So yes, we have done work on that. That was done by Frank Byrne. Imidacloprid was one of the insecticides that were looked at. I don't know if we looked at height so much, but he did look at uptakes into the leaves, and he did determine that. So he looked at uptake into leaves based on concentration of imidacloprid that was applied to the soil. I think it was through the irrigation lines. And he also determined what the uptake level had to be to reach a critical concentration in the leaf to kill lace bugs. That was obtainable. But I don't know how far up into the canopy he looked. He may have just picked leaves around, just the easy ones to grab. So I can't answer that question. The other thing with that is, and I think you guys have a lot more experience than, obviously I do, is that soil chemistry really affects the uptake of some of these systemic insecticides. So I think if there's a lot of organic material, a lot of clay, for example, it's going to inhibit uptake rates. Pardon me? It gets tight. Exactly. Yeah, it binds to that material and it can't move. So that's something else that might need to be looked at when you're considering these soil applications of these systemics. Yeah. So I can't answer that question about height. Yeah. Sorry.
You can avoid that by aerially applying the imidacloprid. From what I've seen, there seems to be 2 different damages, and it's interesting you brought up the disease complex earlier, because the margins I was seeing on some of the damage isn't consistent with insect damage, in my opinion. And you can definitely see the stippling and direct damage from the hemipteran insect in this case at each life stage. You can really see it. And in fact, they don't seem to even move around much on you. I think sometimes because they're just straight attached to the leaf at many points in their instar. I'd be curious to find out how much of the damage is coming from the disease end of it, because I have seen, in correlation with what some other people said in here, more leaf drop where the margin style damage was more evident on the plant instead of the stippling. So it just seems that there may be something going on there disease wise, and it's been hard for me to confirm. It's just a PCA out in the field where, at least hearing it from pathology within UCR, it's something interesting.
Yeah, so I agree with you on that. I think it's important. I've talked to pathologists at UCR. They reckon this is an easy fungus to collect from the field and to run, petri dish assays, do the Cox's postulates and that sort of thing. But I agree. It would be interesting to know what is the driver here? Is it the lace bug feeding, or is it more attributable to the pathogen? Or do you need both of them together to drive this system? So yeah, it would be nice to be able to tie the contribution of each one of those potential agents down to see which is worse. But I still think if you take the lace bug out, there's going to be no opportunity for the fungus to get into the leaves. Yeah, but it would be nice to know the whole jigsaw puzzle there.
All right,
Yes.
we'll do one more question and then we'll take a break for a few minutes.
My one question has two parts.
Okay.
All right. So it sounds like San Diego got hit hardest in like 2017. I'm wondering if there's any evidence that some of those groves have recovered and are better than they used to be.
Right.
That's question number one.
Good
Question number two is
and
I
can't-
saw
I don't know.
on the insecticide list, I saw Bravaria. I'm curious if anyone has tried that.
Yeah, so about orchids, sort of, if you want to call it developing immunity to lace bugs, I'm unaware of any chatter about that. I guess Enrico Ferro would, he was one of the lead guys, PCA down there. Has anybody talked to Enrico about this? Jim? No. So I don't know. What are you still out in the field looking? Do you have any feel as to whether the trees are handling it better than they did originally? No.
Well, I definitely don't see anything like immunity in terms of orchards that don't have any lace bugs. But they do seem to be able to handle it better, like Mark was saying, and I think that might be an interesting study. Maybe it's some epigenetic thing that causes them to, after they get exposed to this, to have some resistance to the damage itself. But I think when we talk about the panel, we're going to talk about a lot more, but I don't see it in San Diego County. It looks like the trees may be able to handle a little better, but they're definitely not immune. And the populations it's pretty much in every grove in San Diego County.
Okay. So to get to your Bovaria question, I don't recommend that either. And the reason for that is we need high levels of humidity to drive an epizootic of those entomopathogenic fungi, and we don't have sufficiently the environmental conditions which would be conducive to a propagation of that type of infection to kill off all these insects. We just don't have it in California. And I'm not the only one to have that negative view. I sound so terrible against my own discipline here. It's like nothing works. But there was a guy way back in the day, 1930s. His name was Prof. Harry Scott Smith. He wrote a big letter, which he published out there, as to why entomopathogens, especially fungi, shouldn't be considered for use in California. And I think his observations way back in the day, after they tried a lot of this stuff, is that we just don't have sufficient humidity for sporulation, conidia formation, and then to drive the infection through these orchards. Ideally, that's how you'd want it to work. You'd want to make one spray, get the fungus established, and then the environment does the rest of the work for you, and you're done. And it just doesn't seem to happen here. Yeah. Yes, last question. Yeah.
So that would increase our relative humidity.
Right.
Would there be a chance that would work on it or improve on that?
Yeah, possibly. So then the other issue with that is temperature becomes important too. So you need sufficient temperature for sporulation and then fungal development, and then you need the humidity to go with that. So often these epidemics seem to do well in warm, humid environments, not so much cool, humid environments. But there are some entomopathogenic fungi that do prefer cool and damp. But then you need those periods for long enough time rather than getting it for a couple of days and then suddenly it's gone and you've got, 80 degrees for the afternoon. And then you drop back down to moist and wet again the next morning. So yeah, that's a tricky set of combinations you got to control. You guys have any opinions on that? Bo?
Sounds right what you're saying.
All right. Thank you. I'll pay you for that later. Okay.
All right.
All right.
Let's
Thank you.
thank Mark. Okay. Thank you guys so much for being here today. Can we go through the panel and just briefly introduce each person? We'll start with you, Tom.
PCA, Oxnard Pest Control.
Tim Buffalo, grower.
Robert Abbott, grower in Carpinteria, organic.
Stuckey, grower in Carpinteria.
Jim Davis, I'm a PCA in San Diego County.
Thank you. I'm going to throw some questions at you, and you don't all need to reply. So just maybe, pick which question you want to answer to. Maybe we'll get 2 or 3 responses to each one. So the first question is, from your field observations this season, how widespread and damaging is ALB in your orchards? Have you noticed particular regions, cultivars, tree ages, or environmental conditions where problems seem worse?
Okay. Can you hear me now? Okay. We've got 20, 25-year-old trees. And what I've noticed is on the perimeters, they were. And that was probably my healthiest trees. On my other field were planted in 04. And they were the weaker trees. That's where I have problems with. Especially it's a cold spot on the ranch, so the trees just have a tough time, and that's where I've seen more damage out in that area, especially where you try to hold fruit, and all the energy of the fruit of the tree will go into that fruit, and so that's been a problem.
I did the same thing. Okay, in my experience, we have kind of an epicenter where it started. We have about 5 acres that are badly damaged. And that epicenter is where the bin drop was. And it's also our headquarters. So a lot of activity there. Also, we're getting a bit of a peripheral effect, like an edge effect. And then from there, it has spread out. We have two parcels. One is down in the bottoms where it's the trees are huge and they want to be huge, and that's where the ALB is the worst, is in these big trees. They're mature trees, and they have more of a jungly kind of growth aspect to them. We have another parcel that's up on a hill with heavy soils, clay soils. There, the trees they're more like bushes than trees, really, and they have they flush earlier, the leaves harden off earlier, and we have thicker, more resilient leaves. And we have the ALB there too, but it's not causing the same damage. And when I speak of damage, it's really the defoliation. It's in the 5 acres that we were affected, it was pretty bad in terms of defoliation. I think those trees are going to have years of work to come back to proper health.
I'm just curious about rootstock effects.
Yeah,
These areas that have high infestations versus lows, is there possibly a rootstock effect here?
Yeah. No. Most of our new plantings are Hass on Toro Canyon. But we do actually have quite a few trees that are the old Topa Topa seedlings. And they are, I will say that there is maybe a correlation that you can draw among stressed trees, whether they're stressed because they have a heavy fruit load, or they're battling Phytophthora, or the soil, it's just a wet spot or a cold spot. We have kind of seen that. It's just one more thing. It's like where you don't know where that threshold is, and you get one more little piece of damage, and suddenly it's a downward spiral. We do have in this same block, we do have some nabals, which are a cousin to reed. And I did notice the nabals were almost like they would harbor a lot of the ALBs, but they wouldn't defoliate. So in a way that they were almost worse because it was like a source for the bug. Yeah. Yeah.
Thank you, Carl.
On a 40-acre block, mostly river bottom land, well, actually all river bottom soil profiles. I noticed no perimeter effect. And it started in a place that was a long ways away from any bin drop. Not to downplay the bins as being a mode of movement between orchards or infesting new areas. In Carpinteria, I'm in a lot of different orchards. One of the biggest differences I saw were orchards, well, in Carpinteria, because of our temperate climate, that's why we're the center of the lace bug. We're also the center of avocado thrips. So it's common that most people spray for avocado thrips late spring, very start of summer. Places that did aerial applications of abamectin and oil, a little bit of oil in it, have the worst lace bug. Orchards that I manage, because of greenhouse thrips, we put in oil with abamectin and spray from the ground at relatively high rates. Or damage is much less. So I think the oil is probably a big control there.
Yeah, I have one comment on that, and I need to go back and look at this in the literature. But Joe Morse with Eduardo Jimenez, they did a lot of work evaluating insecticides for the 2004 infestation. And if I recall correctly, their conclusion was, and maybe some of the PCAs can help me with this, that abamectin wasn't a particularly effective insecticide against avocado lace bug, even though it takes out avocado thrips and probably the mite as well. So having high expectations for abamectin may be unfounded. But yeah, the oil probably smothers them.
Yeah, I reviewed the same literature and with another PCA, and abamectin is not a control for lace bug.
Any other follow-up thoughts from anyone on our panel or in the audience about which kind of regions, cultivars, or environmental conditions have a worse ALB problem? Yeah. Oh, wait.
North-facing slopes, we've seen a heavier population sustain through the more harsh times of the year. So overwintering, oversummering through these heavier heat events. And, I think along with that contributes taller trees and more kind of hedge-grown trees where canopies are interconnected. Those also seem to really sustain populations.
Okay, so our next question, we want to know, what are you doing for scouting and selecting treatment thresholds? When should scouting start? And maybe think about a scenario like Carpinteria, but also in Ventura where it's not a problem yet. Yeah, Michelle.
Well, we scout monthly, and during avocado thrips season, it's sometimes, every 2 weeks. So just from that perspective, we're out there every month at the very least looking. And as far as treatment thresholds are concerned, we start to see some defoliation. We're trying to pull the trigger. However, as we were discussing, materials are limited. Methods of application are limited because of the sensitivity with whether it's greenhouses, cannabis, or the agrobin interface. So we're out there looking all the time, in other words.
Also, the interface, or we haven't even mentioned bees yet. So most of the materials that are allowed for use on avocados and registered for any pest, the majority of them have a bee warning on them in some way, shape, or form. Either the toxic to bees, you can't treat over blooming crops, you can't treat over any flowers, and that includes weeds. Our representative from the Ag Commissioner is here, and I just through conversations scared the hell out of me that we can't use any of these materials when there's a flower out there. We had flowers in January. We've got them all up till this point, and we'll have them till fruit set. So that eliminates the possibility of using imidacloprid, even if it is affected by air, without getting in trouble. So
that's one of our only ones that's not a 3 for its mode of action. So.
And also, I've never used antidepressants before, but after Mark talked, I'm seriously considering it.
Off the first question, I've noticed that the ALB is more prevalent in riparian areas where the humidity is greater. Notice that a potential host is the toyon Christmasberry. I've noticed significant populations on that native species. And then in terms of scouting, I like to scout weekly, especially for groves that don't have presence of ALB. And if it does show up, that it's controlled before it gets widespread.
I have a comment on the toyon. We do have a native lace bug that lives specifically on toyon. So it may not be avocado lace bug.
As you mentioned earlier, can they interbreed?
Probably not these two. No, these are in two different tribes. So I think it's probably okay that we know interbreeding. Yeah, but toyon can hold very high populations of a native California lace bug. It looks very similar, undersides of leaves, similar stippling, little fecal droplets everywhere. Yeah, so I've collected a lot of those. Yeah, so I don't think the toyons. So don't rip out your toyon, please. Keep your native trees around.
Well, I've been mostly scouting for the large dead patch in the leaf. That's very easy to see, and I use it as kind of a percentage. I count a percentage of the leaves that have that type of mark. And while I'm doing the scouting, I also check the leaves to see what percentage of the ones that I can actually reach have live lace bugs on them. So I try to determine, does the leaf have, if it has the dead patch on it, does it also have live lace bugs? Because the lace bugs will leave the leaf alone with that patch on it and have no lace bugs on it if it's an old infestation. And, Mark, I'd like you to correct me if I'm wrong in this, but it seems to me that the lacewing life cycle is that a pair will land on the leaf and begin a colony, and that colony will go through to adulthood on that leaf, but then they leave the leaf and they look for a mate and they find an unused leaf to start a new colony on. And there may be other variations of that, but I think that's the predominant one. And that's why they don't kill the leaf. They don't keep feeding on the leaf until it's completely destroyed, like mites will do or other types of leaf-feeding insects. It seems like once that colony's done, they move on. So I think of those leaves with the dead patch on them as the indicator of that it had lace bug on it, and it may still. And because it seems, from my experience, that once that percentage gets above about 40%, then you're susceptible to the defoliation. And also, it seems like there's some type of threshold level that when they get above that population or that density, they start to multiply more freely, or they get into that exponential phase where, and I think that increases the chance of the defoliation. So basically, as long as I'm seeing, 10 or 20% of the leaves with that patch, I think of that as a non-treatment scenario. I feel like I can live with that. I don't think that they're going to grow that quickly over the next couple months. So I just keep track of them. But when they start getting to that 30% level, then I start really monitoring more closely and checking different areas of the grove that I might normally go to in anticipation that they may reach that 40 to 50% mark, in which case I'm going to want to do a treatment. So that's been the scouting thing I use. And I do look at them all year round, but basically it's mostly that summer period and into the fall where I feel most likely I'm going to be doing spraying, where I most often see that population achieves that level that for me is treatment level.
It's really helpful to hear how you're working through that decision making. Are there any other comments from the panel about selecting treatment thresholds and what you're doing when you're scouting that's been helpful? Or from the audience? All right, let's go to the next question.
I don't think it's seasonal in Carpinteria. I mean, you're in some warmer areas. For example, we had relatively low populations of adults in December, January, but then we hit a couple weeks of 80+ degree weather in January and the population exploded. So in Carpinteria, it's checking every week all year long. There is no start and stop to it.
The next set of questions are about chemical control. So we want to hear, what products have you tried so far and how effective were they for you? How long did suppression last and what timing worked for applications? Why don't we go? Shall I see your brain working? Go.
So I'll respond to that. Because of that population explosion in January, we sprayed the first week of February with oil and Savano. And the control was really good. But we used relatively high rates of material. These are mature orchards, size range from 2-year-olds, emitter plants, to trees older than I am. And not all of them giants, but yes, pretty big trees. And we applied anywhere from 300 to 600 gallons an acre of material, and it was a costly adventure. Yeah, it was all hand spray from the ground. Like I say, we can't. Well, Mark was optimistic about getting good coverage on slow flying helicopter. That's not been my experience. You can't get the coverage on the bottom of the leaves consistent enough. You get some knockdown, but it's not enough of a knockdown to really control the population.
All right, we'll do Michelle and then Tom.
We did some organic applications for 400 gallons per acre, Pyganic and Gavicide 415. And we didn't see very good control. In fact, no residual on that material. Efficacy was low, which we kind of anticipated, just dealing with it over the years. But we're disappointed at the very least. We do all of our hand applications as well at 400 plus gallons per acre, and I agree with you on the helicopter applications. On hillside stuff, it's where you can use a helicopter. We can't sometimes get up on those hillsides with the guys and the equipment, so we might be, that's where it's at, on that type of application. But anyway, yeah, the organic stuff at this point isn't really working.
Tim.
Okay, as I mentioned earlier, we went conventional. We didn't go organic, and we did use the Mustang and Admire. And I did get full control of it. The reason why and I did it by helicopter. We didn't do it by ground. And we did it in 1st of November, late October. 1st of November was the application. Like I said, we did on the larger acreage, you did a perimeter, and that seemed to keep them from moving in. On the other acreage, it was the weaker trees. We had to do a full application to stop them from moving my neighbor. My PCA is contracted to look at my neighbor, and I did stop them from going next door. So that's what we did by helicopter and conventional chemicals, and it did stop them. We did have leaf drop, and nothing we can do about that. So we're flushing now.
I believe that the organic orchard that Michelle was referring to was probably ours, or we were one of them. So yeah, Pyganic did not achieve a great control. We tried MP'd, we tried as a direct. I think we've sprayed 8 or 9 times since last July, and we're still fighting it. So we've actually shifted our philosophy away from pesticide applications, and we've started using materials that just create an inhospitable environment on the leaf, including clay. So this is like Surround WP. We've used silica sprays. There's a blue gold product which has silica in it, and it's a surfactant. It's with micronized silica. And the last spray we did, we threw some molasses in there, and we also threw some nitrogen amino fertilizers in there to kind of create this kind of sticky, sharp, inhospitable environment. But we're also, so the leaves are white, some of the fruit's white. I don't know how it's going to pack out, but we're not seeing a lot of insect activity on those leaves, bad bugs or good bugs for that matter. So we've, I guess we're on the dark side now.
To follow up with that, have you tried those strategies independently, or did you kind of just do them all, at once? Have you been able to kind of suss out which one you think is working the best?
I'm gonna say the silica is the most effective. Yeah. And and that is kind of an opposite approach to oils. So if you're going to use oils, you don't want to use these drying materials.
That's a great transition to our next question, which is what other management practices have you tried? Have you observed predation, for example? For cultural control, have you tried pruning, cover crops, things like these silica sprays? What have you guys what are the other experiences with that? And Tim, you had a thought too.
Wait, okay, so we do prune, and we prune about every other year, and it seems like to keep the trees down, and it keeps them in control we keep them about 15 feet. And if we don't do that, then they kind of get out of control, and you get a lot of water sprouts coming up, and then you have all this extra foliage. So, and then we're pruning just on the sprouts and not the fruit wood. So we're going through and doing that. I had a follow-up question here for Robert. Have you tried Sabadilla? That's just with maybe molasses and sugar or something like that. Has anybody tried that material?
Will you repeat the question into the microphone? People are having a hard time hearing.
I was mentioning Sabadilla. Have you tried that material? It's a poison. They have no.
No, I don't think so.
That's an old one we used for thrip when we didn't have the agromech, and we flew tons of that stuff out, and it seemed like it worked okay sometimes. Sometimes it didn't work okay. You can't get Sabadilla?
Yeah, that's right.
Michelle says it's too hard to get Sabadilla now. Yeah.
Sugar.
Plus you need sun and, May, June, May gray and stuff. It works when you have sun, but you can't get it right now.
Well, I've pretty much used all the registered insecticides, and conventionally, pretty much all of them would work. There's really 4 major ones: Savanto, Delegate, Imidacloprid, and another one that I can't remember at the moment, but I'm sure I'll think of it in a second. And all of those have worked very well. I've done all my applications by air, since in San Diego we don't have much avocados on flat ground, so we pretty much do all our applications by air. But on the organic side, none of the materials have proven to be very effective. The most effective one, I think, is Entrust, and if you get good coverage with Entrust, you can get good control. But the problem is getting the coverage by helicopter. Now, one of my biggest clients was Henry Avocado, and they like big trees. So they had 30, 40, 50-foot trees. And no matter how slow you made the helicopter go, we could not get penetration down all the way down. But if you have trees that are 20 feet or less, I think you're probably going to have very good coverage with a helicopter at 75 to 100 gallons an acre. Like I say, I've had good luck with all those materials. Now, one of my most challenging grows was a 60-acre organic block that had a persistent problem with the lace bug. And I should say that in my experience, the areas that are most likely to get lace bug were north-facing sides of slopes. So to me, that means cool and more humid. So those might be the things that are causing them to be on that slope. So you guys may have different experience up here, but that's when I scout, I always check that side of the grove because that's where I get the most finds and the most populations and the most spraying. So this grove I'm about to describe, it was a east-west grove, a hill that ran east-west. So I had an entire side that was on the north. And that place was just intractable. I sprayed it every year for 4 years with Entrust one time, and I could just sort of keep it down. But on the 5th year, the grower wanted to try to get complete control of the grove because he didn't like the results that we're getting with this partial one. And so I sprayed the grove with Entrust with a helicopter, and then I came back in and sprayed the bottoms of the trees by ground with Entrust with a ground rig. So basically that cost him, it was $600 an acre for the ground rig and $300 for the helicopter. So $900 an acre this guy spent to control the thrips, but that did the job, right? That actually solved it. So I think if you can get good coverage, if you have small trees and you do it by ground or by air, you should get good results out of entrust. The Pyganic didn't really last very long, and so I think that it didn't really get the adults very effectively. So I haven't been recommending it. But really, I think we need to do more research on the organic products, and possibly there's some combination or something that, where you're using like an oil that agitates them and gets them to move around so they pick up the pesticide more easily, or something like that. But we really need something on the organic side.
Thank you. So yeah, I'm hearing kind of repeatedly that conventional products are working, we're lacking organic options, and that pruning might be really important for keeping trees small, ensuring spray coverage, and then also spraying creatively to get under the leaves and on top of the leaves fully.
I want to, one thing about pruning is we have a block that is kind of a juvenile block where it's starting, the trees are starting to crowd each other. And I really like to keep the lower leaves, the skirts down when I can.
Mm-hmm.
But in this situation, we were not achieving good coverage because there were a lot of leaves that were close to the ground and we just could not get inside there. So we did end up pruning up the skirts, and we ran our flail over those prunings to destroy all the bugs. And so the leaves that were closer to the ground had by far the most amount of avocado lace bugs on them compared to higher up in the canopy.
Ben.
A Zoom question. Has anyone tried a sail to control ALB? Or a sail?
What's the active ingredient in that? Do you know what the active ingredient in a sail is? Oh, it is. Okay.
Any thoughts from the audience, Derek, about either chemical control or non-chemical control options you've tried?
So I know they don't stump down south anymore, but I'm curious if you stump a tree, break it for 2 to 3 years, if that cancels the life cycle or helps.
It doesn't seem to really, at the beginning of the year when the new leaves come on, they're not on those leaves, but eventually they do move on to them. So yeah, it does prevent them from being on, but only for a short while. They're not great flyers, as been mentioned. I mean, they can fly and they can fly effectively. It just seems like they don't fly very often, or it's hard to motivate them to fly. But they definitely can fly, and they do get on the stump trees, usually within the first year after the foliage comes out, but if not, usually by the second year for sure. So I think pruning to do height management and canopy management helps because of the spraying effectiveness, but I'm not sure that it really influences the pest itself enough to be a management technique.
Derek, you make a good point. With a shorter tree, a more vigorous tree, you might be more successful getting uptake of an imidacloprid product. It would act more like a young tree, so that's worth looking into. And just a thought about coverage, when it's all relative. When I started in this industry many moons ago, good coverage on citrus that had good spaces between it was 12-foot high citrus tree was 1,200 to 1,600 gallons per acre. We're trying to get good coverage on avocados, or claiming we're getting good coverage, and I don't think it's possible. Avocados were not planted with the thought of we're going to have to spray these. Originally, they were planted on hillsides and put real close together, and we let them grow to enormous heights. And I don't see it with helicopter applications. It's either either we're thinking we got a good coverage, or we're not looking close enough. So now that's with contact materials. When you have an imidacloprid product that can be absorbed into the tree and get movement, and also spirotetramat, which is trade name Movento, there's 2 other materials that we could try. I don't like even mentioning the trade names here because each situation is different. Each grower tolerance for damage is different. If you've got a new client that just has the start of ALB, you better be cognizant of the fact that they may not want 0 is the best program for them. So it's a case by case basis, and we've had orchards that defoliated quite significantly. If you've seen the video, that's one of our orchards. So we didn't want it to get to that point, but we also were able to try different products and see what would happen or what didn't happen.
This is a question for the carp growers who use the conventional methods. How quickly did you guys see the effectiveness? And then how many times did you guys have to apply?
When we, this February application with oil and Savanto, it was effective almost immediately. Kill was, you come back within 2 or 3 days and kill was pretty much complete. I don't know if there will be much residual effect from it. And I haven't sprayed other than our June or first week of June, end of May spray for scrotothrips, where we've applied oil and abamectin. That's the only other spraying I've done.
Yeah, the only thing, yeah, I do inspect it once a week, and I scout it to make sure we're fine. But the spray is probably this right now. We did spray in last year, November, like I said, 1st of November, and I haven't seen any ALB at this point. So I've seen other things, but no ALB, and it does hold it for a while. So if that answers your question.
Next question is, what limits your ability to manage ALB effectively right now? What limits your ability to manage ALB effectively right now? Avocado Lace Bug. Oh, ALB itself. Robert?
Well, okay. It's like, well, that Tom was mentioning too about the bees, and we kind of neglected that. If we have to spray something, I move the bees out, and we give notice. And if it's that serious, they've got to go, and that's kind of how we have to manage it at this point. And I'm just not really looking forward to this year for the ALB. Like, we don't have to spray. Like I said, it's just economic hardship on us, and we're looking at numbers right now, and just like the numbers don't look good, and with the price of avocados right now, it doesn't look good. So that's kind of where we're at.
Well, my approach is basically I'm constrained by the like Tom said, the individual growers' damage, perception of damage, or that their tolerance of damage, and also the biology of the pest, and also the economics of the situation. So in the years since 2017, when we've had this pest, I've never sprayed a grove more than once in a year for the ALB, unless it was an organic grove, because like I say, these conventional products are pretty good. So I don't think you're going to need to do multiple sprays with them. I think if you have a pretty significant population of ALB in the spring, and you're going to do a thrip spray, that if you used a material that also hit ALB, that would probably be a good time to use that material, especially if it does both of them with one material, like one of the stronger materials. But otherwise, I'll make a separate application later on in the year if it warrants it. So basically, I think that the things that constrain me is the pest and reach the treatment threshold, and and that threshold varies a little bit depending on the grower's risk profile. So and then also, I don't want to spray multiple times. So I don't want to spray at a time when I think that it's likely if I spray now, I'm going to have to spray again in the near future. So that's what constrains me.
Just to add to that, if we're tank mixing, like you were saying, Savanto Prime is a bee-friendly material. So it would be something to add to your Agromec spray or abamectin spray. When we're talking about multiple applications, if you're doing a hand spray by ground, it can get very pricey, like you were saying. So if you're going to tank mix, that would be a good time to do it.
Yeah.
I have two questions. One is for the residue organically with silica or the surround products. Have you seen a positive impact with sunburn from those products? And hopefully somebody from the audience can comment on pack outs and how that's going to look. Second is with doing multiple applications of Entrust, do you time that at the same time as your thrift sprays? There's documented evidence of resistance with Entrust and thrips. If we're having to spray multiple times throughout the season, are you worried about resistance, or are you pretty confident that you can rotate with other materials?
Well, I'll address the second part of that question first. I try not to use the Entrust more than once a year. I think the label allows 2 applications. But I try to limit it to once a year because of the resistance and also for the cost. It's very expensive material. But the problem with that is that we don't really have a rotational material organically. Like I can't really rotate the Entrust with Hyganic because the Hyganic's not effective. So basically, as I said, would do back-to-back sprays, but just once a year. You know, and I'm hoping that that's so infrequent that they don't get resistance. But I think that's another really strong selling point for doing research to find organic products that we can rotate with to keep them all effective.
Thank you. Okay.
For the clay and the silica, for sure. I love those. I love those products, not just because they create this environment that the bug doesn't like, but there is for sure some sunburn protection. I've also seen it's a growth modifier. I mean, it's a physical thing. You're slowing down the transpiration of the leaves, and that's going to, it's the same effect that you would get as if you put shade cloth over your entire orchard, and that will encourage fresh growth. So we've used it in the past, not just after heavy pruning, but to stimulate fresh growth, and I'm a big fan of those products.
One question back there.
So, I think Tom's point with our labeling is very critical, and it's something I've thought a lot about with treatments I've done down in San Diego. But I guess this might be directed a little bit towards Mark, but is there a critical life stage point within these population growths that would be more effective to treat when there's more instars of actual live bugs on the plants instead of eggs? I think I've seen that a little bit down into like fall time going into winter when there's going to be a lot of overwintering, and there's not many eggs left in the population. Seems to crash them a little bit faster because they can't rebound quite as quickly, but I just don't know if there's something that we could find out there.
Yeah, so that's a good point. I'm actually working on that big data set now to try and figure out where the fluctuations are most, I guess where the bottleneck is for those different instars and the adults. But I kind of like this idea. Maybe these guys who have some experience, we call it dormant sprays, where you go in during the winter to try and knock back these overwintering populations. And I was wondering if anybody's looked at that for avocado lace bug. You go in when the population densities are low, probably not much bee activity, very little flowering activity going on in those orchards, and trying to whack it. Then has anybody looked at that?
I have sprayed a few like that, Mark, and it's worked well for me. I looked at it as and the imidacloprins, I could either do one really effective spray, or I could split it into 2 doses based on labeling. And it was more effective to wait until there was more bugs on the plant in terms of live bugs than eggs. It did seem to work in a few of the growths. I did.
Yeah, so that's a really good point too. I don't know if we've got any good efficacy data against eggs, with any of these insecticides. Does anybody have familiarity with whether or not these insecticides have any impact against lacebug eggs? We've always looked at nymphs and adults in these bioassays.
I'm sorry, they had an impact on what?
Yeah, on eggs, on lacebug
Oh,
eggs.
yeah. I haven't seen really much impact on eggs.
Yeah, okay.
From any of the pesticides. The hatch seems to be the same, but of course the young ones that hatch out are the most susceptible. So if there's any residue left, they die pretty easily. Also, I've seen from the heat that the young instars are the most susceptible to high temperatures. You see it first in them. And actually, I haven't even seen it get hot enough yet to kill the adults, at least not a lot of the adults. I've seen adults survive all the heat waves we had so far. So the young ones, it definitely kills them off. But the bigger ones and the adults are less susceptible to that.
Let me throw the last question out there and then we'll open it up to more questions and discussion. So if you could have answers to one of your questions tomorrow to improve ALB management, what would that question be? For example, what is another organic control option? Could be a question that Jim has brought up repeatedly.
I think the last point of where is that inflection point of the best time of year to achieve control?
My question would be, when are we going to get an effective, cost-efficient biological control of ALB? Which would mean Mark would have to go somewhere in the Caribbean, most likely.
Oh, he said Western Mexico. Such far.
Western Mexico.
Yeah, West Mexico, yeah.
I volunteer for you can just take an entire avocado tree and drop it on top of our property from Mexico. I'll take all the critters that come with it.
Oh, I don't know if you want that. I've seen some pretty bad stuff down there.
Tim,
I'd really urge everybody to do soil tests where you take an FGL or soil and plant lab. Make sure your soil is strong. I use organics such as humic acid on it, and it seems to really strengthen the tree. So I think soil fertility is probably number one you need to look at.
tree health, soil health. All right, from the audience, what is the burning question you have about ALB that you want answered right now?
What can we as the industry do here that are currently unaffected by ALB do right now to prevent its spread?
Mark, are you writing all these down?
Spread.
This is recorded, so.
Yeah, the questions that interest me, yes.
I see Ben back there. Oh, sorry.
Not respond to that. No, sir, don't come my way. Well, on that question, I'm curious if anybody is mapping the spread of lace bug and trying to keep, that's one, it'd be helpful to know where it's going. It's been in CARP for quite some time now, getting into Goleta a little bit. It was news to me that it's been in Orange County, but I'm curious if there's any information on use of plant growth regulators or insect growth regulators, which I'm really unfamiliar with, in combating this one. And then, I guess while I have the mic, we've got a very interesting case study in CARP, one of our neighboring groves, really well managed, smaller acreage, top-notch grower, doing the soil samples, leaf analyses, and got the nutrient levels just right. Almost no lace bug population or damage and leaf drop there, and right on the other side of the highway, other side of the creek, night and day. So
there you go.
Be good growers as much as we can. I think there's a lot to be said for that, and maybe specifically potassium levels. I'd be curious to hear from others more on that. But yeah, question about plant growth regulators, mapping, and grow good.
I see a question back there from someone who hasn't spoken yet.
Hopefully I don't get in trouble with this question, but can the ag commissioner from a county establish a quarantine that would limit or allow us to use imidacloprid during bloom? Does the Ag Commissioner of the county have that kind of authority as opposed to the state?
I don't know the answer to that. There was somebody from the commissioner's office here, right? Oh yeah, David.
I don't use that stuff.
Not my specialties area because I'm not in the pesticides unit, but typically this stuff has to go through a special local needs with like DPR to establish, can you use that product in a different way that's off label? So it's not really our department, but DPR as a whole can be. During bloom. Yeah, I can't speak on that one, to be honest. That's sorry.
I'll comment on it. If you want bees in the county where your orchard is, you can't use imidacloprid. Beekeepers do not want their bees in an area where there's been imidacloprid used during
I don't
bloom.
want that. I don't use it at all. That's a no.
Can I? Yeah.
I see.
Can I, since I have the mic, back to Derek's
I take care of my
question.
bees.
I kind of wonder has anyone identified a pathway that this might have gone into commercial orchards? Because I understand how, it could come from Mexico to a backyard grower, but if it doesn't fly very well, was there a way this came on equipment or new nursery stock? If anyone has idea of how to protect themselves, their own orchard from it coming in.
Mark, do you want to comment on that?
Yes, so we've seen this with Persiamide, avocado thrips, probably true for avocado lace bug too. Points of entry into your orchard, possibly through packing bin drop-offs, are probably high risk if they're coming out of people's orchards that already have lace bugs and those bins haven't been decontaminated. You could inadvertently introduce adults, maybe even leaves in the bottoms of those bins which might have lace bugs on them. The issue then is, how do they get from those leaves in the bottom of the bin up into your trees? But as we've heard, if you're walking through your orchards with a blue or yellow colored sweatshirt, you might be inadvertently picking them up and you move to somebody else's orchard or wherever, they'll just hitchhike and jump off. But yeah, it's a good question. I don't think we really know. That would be the truly honest answer. But I guess I would preface that or qualify that by saying I think humans almost certainly have been responsible for moving it over big areas rapidly and into new zones where it didn't exist previously. So whatever we can do to mitigate those unintentional introductions into new areas would be worth it.
We're going to do one very last question back there.
I've got a question and an observation. My question is on cultural practices. I think fine-tuning our nutrition, maybe like Ben had alluded, is going to be something that could help us. But I'm wondering also on the pruning side, sounds like trees that are at a height for proper application is helpful. But what about just general pruning practices? Do we want to send the signal to the tree to bloom or to flush heavier by making more cuts, or is that counterproductive to building lace bug populations? And then an observation to share was walking through a couple of blocks with a grower who farms both Hass and Gem. And Dr. Hoddle alluded to the fact that the Gem doesn't appear to be a good host for the ALB. The observation that we made is that the Hass leaves tend to grow a little bit more flat and are a little bit more, the underside is more protected from direct sunlight, where the Gem leaf tends to cup a little bit more and the underside is a little bit more exposed to direct sunlight. And I was wondering if there was any thoughts or if that observation might be something that is keeping the Gem a little bit more protected from the infestations.
Mark, any ideas why Gem might be more protected?
No, I don't know why. And those observations are just observations. If we were to look into this properly, I'd want to try and breed lace bugs on leaves of these different varieties to actually have some numbers to present rather than us just going, we looked at them for 2.5 years and didn't really see much activity on them. But I think, you know, if a lot of people are seeing a similar observation of gems where their hasts are infested and then gems that are growing, you know, pretty much right next to them have very little ALB activity, that I think is suggestive that there's something important going on there. But what the mechanism is, I don't know, leaf cupping, leaf thickness, whether there's secondary plant compounds in those leaves that ALB doesn't like, or maybe it doesn't recognize it as a host. I don't know.
I'd like to comment. Is lacebug negatively phototrophic? It does sit on the back of the leaf, so is it adverse to sunlight? Yeah, it starts moving. You flip that leaf over.
Yeah,
they look like it to me.
And that goes back to Giuseppe's comment about the curvature of that gem leaf. It may be less hospitable to a lace bug that's not liking sunlight because there's not that flat aspect that a regular hast would have. I think that was a good observation.
Yeah. I don't want to end the conversation because it's really productive, but I wanted to get you guys out of here at 11. So what we'll do is I'm going to just volunteer all the panel to stick around for a few minutes if they can to answer more questions, maybe just one-on-one if you can. And I know Mark and I will be here as well too. Thank you all for coming today. It was really awesome hearing all your thoughts and ideas.